As to me, I am Dr. Ajay Kumar Patra, former Emeritus Professor of Zoology, Utkal University, Bhubaneswar, Odisha, India. I am an environmentalist as well as a scientist. Basing on my 40 years of research experiences, a well-planned scientific methodology, plasma serum therapy has been postulated against human corona-19. This therapy for sure will help to synthesize vaccine against novel pneumonia COVID-19. I will be happy that let the scientists of the world discuss, criticize my methodology in detail and let them help to synthesize vaccine against novel pneumonia COVID-19 as soon as possible. I am just going to narrate the basic mechanism of my newly invented plasma serum therapy as follows. First step, to begin with, I have classified COVID-19 patients into three groups as such: mild stage from first day to 6 days of incubation period, critical stage from 7 days to 11 days of incubation period, most critical stage from 12 days to 14 days of incubation period. Besides, one healthy man is kept for control group. In each group, there will be one adult male person of more or less equal age group ranging from 40 years to 45 years. Each group will be named as for mild M1, for critical C1, for most critical MC1 and for control CN1 respectively. All these four groups of experimental persons will be kept isolated from each other under strict medical care. Only one doctor, one nurse and one attendant with all health precautionary steps will be allowed to observe them regularly, at least thrice in a day. Second step, let us now come to the research laboratory. Since I am a zoologist, we usually start research with lower animals. Here, with prior permission of ethical committee, 12 Indian captive healthy male rabbits are procured and are kept isolated in the research laboratory, dividing four groups. That means each group comprises of 3 male healthy rabbits of more or less same weight groups. All these rabbits are acclimatized in laboratory conditions initially for 7 days, being fed with healthy food, for example fresh morning grass and grams freed from any contamination by a single attendant only. In this case, one scientist, one researcher and one attendant with all health precautions will observe separately all these rabbits and record minutely the change of behavior, if any. Again, after 7 days, all these four groups of rabbits are acclimatized at 20 degrees centigrade in the laboratory with proper feeding and caring as mentioned above. Since the deadly human coronavirus 19 has originated from the wet market of Wuhan city in Hubei province of China, where prevailing environmental temperature is below 20 degrees centigrade. Only one attendant with all precautionary measures will take care of all rabbits All these 4 groups of rabbits are named as for mild M2, for critical C2, for most critical MC2 and for control CN2 respectively. Third step. Let us now go back to 3 groups of patients along with a healthy man. After keeping them 6 days strict observations as per their incubation period, blood sampling of about 50 mg will be taken from the vein of the right hand at early morning with fully sterilized hypodermic syringe. Usually, the scientists do prefer early morning for sampling because of low temperature, coolness of environment, and especially then the basic metabolic rate BMR is low. Before collection of blood, all sterilized instruments, test tubes must be washed with anticoagulating agent like Ringer's physiological solution. While sampling, the precautions as per the guideline of World Health Organization for novel COVID-19 must be maintained. The blood samples are then processed in the laboratory for preparation of blood plasma. Blood minus corpuscles or liquid blood is called blood plasma, and then from blood plasma, blood serum, that is blood plasma minus protein fibrinogen is called blood serum, and that blood serum will be synthesized. Those 4 groups of blood serums are nothing but the antigen of human coronavirus 19 and are kept separate at low temperature above 40 centigrades and below 20 centigrades with care. After naming them as for mild M3, for critical C3, for most critical MC3 and for control CN3 respectively. First step, sampling. Let us again go back to experimental rabbits in the laboratory. Dose. The dose for treatment to rabbits must be followed as per the prescribed dose of World Health Organization or begin from lowest dose 0.5 mg of rabbit or maintain trial and error methods. Administration 0.5 mg dose of synthesized serum antigen prepared from man will be injected to only one experimental rabbit at intramuscularly in early morning. The reasons mentioned earlier. The rest of the 2 rabbits of each group will be kept aside for future experimental use. That means M3 to M2 body, C3 to C2 body, MC3 to MC2 body and CN3 to CN2 body. First step, all above experimental rabbits will be kept for 21 days at 20 degrees centigrades under strict observations. During this experiment, the rabbits will be fed liquid food, for example glucose or fructose, orally in the morning time only. They are kept isolated. Their lethal dose 50 if taken more than 3 in numbers at a time. All behavioristic patterns of rabbits under treatment will be recorded for time to time. Antibody formation. The serum antigen of affected man inside the body of the treated rabbits will soon start working. The leukocytes and monocytes of WBC and also B cells of lymphocytes of WBC will work together against administered novel COVID-19 serum antigen and do enhance the immunity of the rabbit body, forming more and more antibodies from serum antigen through their phagocytes. Their survival values will also be recorded. Sampling. After 21 days keen observation, the sampling of venous blood will be collected from treated rabbits, also untreated one, and be processed as per above rule to synthesize first blood plasma, that is liquid blood, and then plasma serum, that is blood plasma minus fibrinogen. The plasma serum synthesized separately from 4 groups of rabbits will be collected and be kept under less than 20 degrees centigrade with care. The samples are named as for mild M4, for critical C4, for most critical MC4, and for control CNO4. Following the above methodology, we can also start with adult women ranging from 35 years to 40 years of age and children ranging from 10 years to 15 years of age, and accordingly antigen serum will be synthesized. These antigen serum will be stored at very low temperature, less than 20 degrees centigrade, for further preparation of vaccine or antibiotics against deadly human coronavirus 19 novel COVID-19 disease. Conclusion. This plasma serum therapy for sure will help a lot to synthesize vaccine or antibiotics against human coronavirus 19 if necessary with little modifications. From this antigen serum, we can synthesize vaccines separately for children, for women, and for men as well. Also, distinct vaccines for mild stage, for critical stage, and for most critical stage are possible. Those to be formulated as per World Health Organization or through trial and error methods. If rabbits survive, then this antigen serum therapy will be for sure successful in case of persons suffering from human coronavirus 19, since the biomass of rabbit is far lesser than the biomass of human being. I welcome criticism, discussion, and critical reviews of my work and appeal to the scientists of the world to practice my methodology to make it success in synthesizing separate vaccines against human coronavirus 19 for children, women, and men, and thereby we can save the world people from corona panic. Joy Hind.